Welcome to the presentation. My name is Fazar Irfan and I'm student of Master of Medical Biotechnology. I will be representing the analysis of quantitative data of E. coli sample in order to infer the unknown treatment. The two software which are used for data analysis is Panther and Strings. Proteins and their genes have been classified according to the family, molecular function, biological process, and pathways in order to facilitate the analysis in Panther. Strings are database of known and predicted protein interaction which include physical and functional association. These two databases are one of the suggestion of data analysis. They are very easy to use with good visual representation of the analysis. Panther uses literature by expert biologists to infer biology, and String uses the previous knowledge in databases, high throughput lab experiments, genomic context prediction, automated text mining, and co-expression. In Panther, genome statistics of E. coli shows presence of 4,313 genes which are classified according to molecular function, biological processes, pathways, protein classes, and cellular component. And it is a good reliable number from a statistical point of view. The statistic used in String is also reliable because it uses the confidence level for the number of interaction with highest confidence number is greater than or equal to 0.9. The quantitative data analysis through different online databases, it has been found that three proteins are upregulated, which are 2-methyl citrate dehydratase, 30S ribosomal protein, and 50S ribosomal protein. And plenty of the protein which are related to different metabolic processes has been downregulated. It has been inferred by comprehensive study of recent literature that the three proteins upregulated associated with methyl citrate cycle and ribosomal protein, which are structural constituent of ribosomes involved in translation activity, are related to aerobic conditions. Genes which are related to the upregulated proteins are analyzed and visualized by Strings. Methyl citrate pathway is one of the upregulated pathway, and E. coli is able to metabolize propionate through methyl citrate pathway aerobically. It has been confirmed that E. coli is able to use propionate as electron and carbon source under aerobic, not in anaerobic conditions. Propionate is a volatile fatty acid and its accumulation in cell can block metabolic pathways, which is essential for cell growth. This also supports our finding that we have analyzed that lots of metabolic pathways was downregulated in treated cell, which include amino acid metabolism, glycerol metabolism, and glucose metabolism. The gene which we found in our analysis is PRPD, which is a part of PRP gene cluster, and it is essential for propionate to pyruvate oxidation through methyl citrate cycle. The two other proteins which are upregulated are 50S and 30S ribosomal protein, which are involved in translation. These two proteins are a subunit of bacterial 70S ribosome and provide a platform for amino acid linked together into polypeptide chain. 30S ribosomal protein ensure correct positioning of mRNA, and 50S ribosomal protein has active site for forming protein peptide bond. It has been proved by a study that when changing the conditions of E. coli from aerobic to anaerobic condition, these two proteins had downregulated, which gave us a strong evidence that these specific proteins are related to aerobic conditions. This is a graph between the up and down regulated proteins and group ratio. These are all the proteins which we found related to our treatment, which is aerobic condition. As you can see, three of the upregulated proteins which have group ratio greater than 2, and all the downregulated protein have group ratio 0.5 or less than 0.5. In this table, you can see all the protein IDs, group ratio, and pathways associated with all the proteins which we have analyzed related to our treatment. And the one which are highlighted with red are the upregulated one, and all other are downregulated one. The downregulated proteins, two most important one are glutamate decarboxylase and lysine decarboxylase. They are related to the E. coli acid stress system, and it has shown us that the alkaline conditions are in E. coli cell, and it has also been found in one study that pH effect on gene expression is mainly due to the influence of environmental factors. Most important is aerobic condition. So we can say that the aerobic conditions in E. coli cell has changed the pH of the E. coli to alkaline. There are some other proteins which are related to anaerobic respiration have downregulated, like fumarate reductase iron sulfur subunit, anaerobic glycerol 3-phosphate dehydrogenase subunit A, B, C, respiratory nitrate reductase 1 beta chain. They are all downregulated in our treatment, and they are related to anaerobic conditions. Cold shock proteins are also in response to some environmental stress, one of which is oxidative stress. But in our treatment, they are downregulated, which means it is not oxidative stress. So I conclude here that after analyzing all the pathways, proteins, then we can say that the treatment of E. coli is aerobic conditions. Some of the downregulated proteins are related to acid stress system of E. coli, and it can be inferred here that because of aerobic condition in the cell, the pH of the treated cell has changed to alkaline. Thanks for your time. Bye.